Okay, no.
And
Thank you.
we proceed to the next speaker, who is Adam Williams from University of Minnesota. He will be talking about lunar agriculture, transformation of lunar regolith into soils to enable space agriculture. He's a PhD candidate and professional engineer with 15 plus years experience in test and product development. And his current research is focused on lunar agriculture. Adam, please.
Hey, Nicholas.
Hi.
Thanks for having me back. Yeah, I'm here to present. Can you guys hear me all right?
We hear you well, and the screen is coming up. Yes.
Okay, thank you. So yeah, I'd like to thank the Moon Society for having me back. I'm here to continue to update on my PhD research on astropedology, or the formation of off-Earth soils, in particular how to transform regolith into soil, and the results that I'm getting. So already got a bit of background from me, but yeah, I'm a professional engineer. I'm currently working as a contractor for NASA on lunar projects. My background is primarily in research and development and advanced products. My education spans economics to engineering to bioinformatics to space resources, across over a decade of higher graduate study. When I talk about my research really has four primary goals that are part of this larger effort to develop and characterize methods for transforming lunar regolith into something like soil to support an off-Earth agricultural system. And when we think about it, there can be like four areas of change. There's biological change, chemical change, physical change, and structural changes, and we characterize them all. While the Moon to Mars architecture and Artemis plans are currently undergoing a bit of a rethinking, this is last year's kind of view of the plan. In short, right, I don't need to speak too much to this audience, I think, but we're going back to the moon, this time to stay. There's lots of active partners, some in cooperation and some in more of a competition, private companies, that kind of thing. And there's going to be ultimately a demand for this. And really, when I thought about this, I think there's a critical gating problem, and that's food is arguably the most difficult gating factor for human space habitation. People eat around 4 pounds or 2 kilograms of food per day. This comes out to a massive amount of mass over the course of a year. With our current rocket architecture, we can only support maybe a dozen people on the moon, and that's if everything was going to supporting this habitation, this outpost, right? So we've probably heard of the tyranny of the rocket equation. The farther you want to go, the more fuel you need to bring. The more fuel you need to bring the fuel to go farther. Humans are chemical rockets in their own way. For all the food we need to bring, we need more fuel, and ultimately there comes a narrowing point where it just simply becomes uneconomical and technically unfeasible unless we can sustain ourselves out there. A little bit more about the nutritional needs. Astronaut data from NASA over the various in-space programs have shown that astronauts need just as many, if not more, calories and nutrients here on Earth. Part of that is because astronauts are expected to do so much physical training in space to offset the negative effects of muscle loss and that kind of thing. So calorie needs are just as many, if not more. There's also psychological needs that get talked about, but maybe not as much. Humans are co-evolved with plants. It's not just a matter of we need food to eat. Astronauts are well known when they come. I've talked to probably half a dozen, and when they come back from Earth, they all say they're very interested in plants. They're very interested in grass, that kind of thing. There were experiments from the Russian Soviet era that showed that plants in an isolated facility dramatically improved crew dynamics, just having them around. And then there is a small little greenhouse on the space station. The flowers are incredibly popular. Going and taking care of the plants is seen as a reward, and is known. And it's not uncommon for people to just kind of go and hang out with the plants, particularly if they've been up there for a while. So we really need them to feel healthy. So kind of how do we solve this problem then, right? So the conclusion I came to is we need to use lunar resources to shorten the supply lines and ultimately build off the regenerative techniques that we see taking place here on Earth and leverage biology to reduce costs and equipment needs, which in turn will develop multiple revenue streams that can support products back to Earth and dovetail those same technological improvements back to Earth, right? So an example, right, is we might produce a liquor on the moon that we could ship back to Earth, and yeah, maybe it's $1 million a bottle, but someone would pay for it. Presumably, people pay just as much for rare wines and things like that found from shipwrecks. So it begins to develop an economy. There's also environmental needs, right? The regenerative technological needs, the pollution, the radiation management, really developing closed loop systems. Even those that are semi-closed loop on Earth are not particularly good at really monitoring everything. Things like air gets taken for granted, for example, and ultimately increase yield and improve soil health. So how do we take what's on the left, which is the Apollo footprint, and move it to what's on the right? And I chose the picture on the right very particularly because the minerals that underlie the areas we see in the basaltic tropical areas of Earth are the same minerals that we see on the moon. So these very much could become that. And my research is showing that. Just a little bit before I dive into the results. So the two types of main categories of minerals on the moon. We have the highlands, which is the lighter part of the moon, and then we have the mare, which is the darker part. The current Artemis missions are set to go to the South Pole, which is predominantly highlands, and there are pockets of mare in the South Pole, but it's almost very much the highland composition. This is important because all but one of the Apollo missions actually went to the mare. So our understanding of the geology of the South Pole is maybe not as strong as people might think. My research is primarily done with lunar simulants. Sometimes I get questions around that. I've used the Exolith Labs versions. Just wanted to put that in here for people to reference if they're interested. But yeah, so the highland rock and really the mare basalts are primarily plagioclase, feldspar, pyroxene, and olivine. And what differs between them is the ratio. Mare can also have ilmenite. Ilmenite is generally seen as a potentially valuable resource because it's extremely high in titanium. So in many ways, it could be easier to get titanium on the moon than iron, depending. But the highlands is 90% plagioclase feldspar with pyroxene and olivine, whereas the mare is primarily 54%, give or take, of pyroxene and 80% plagioclase feldspar and then olivine. What I have on the right here is a mineral weathering chart, and what we can see is primary minerals. These are minerals on the left column that are kind of the raw rocks that come into existence. And then on the right is secondary minerals. These are the things that we tend to see all over the Earth because the Earth had weathering and life for many, many years, whereas the moon we don't. We don't see any of these processes. The weathering is driven by wind, it's driven by water, it's generated by freeze-thaw cycles, it's generated by via biology, and really the combination intersection of all of these things. Right, temperature and humidity could make one rock go one way and the other. But I've highlighted here the olivine, the pyroxene, and the feldspar. I've also highlighted the volcanic glass. The moon is known to have glasses from impacts. Though my research, I've excluded the glasses as it generates a lot of complexity. So primarily focusing on these minerals, we could maybe see the olivine and the pyroxene move into amorphous hydroxide oxides and maybe down to something like a goethite or a gibbsite. But really what we're expecting to probably see is the feldspars to weather into dioctahedral smectites and then ultimately probably into a kaolinite, which is a much more the way we see it here on Earth. I do get some questions on whether we can grow plants on the moon. So in this photo here, this is from the University of Central Florida, Annalisa Paul and Rob Furl. Their research, they actually grew plants in actual Apollo samples, and they compared simulants, which is what I'm currently working with Apollo 11, 12, and 17 samples. And the plants all grew, many of them sprouted. The plants were harvested relatively early to begin to see their stresses, but this proved that actual samples could grow. Early plants were exposed in the Apollo area to regolith to see if there's any toxicity. That's the photo here from their research. This is from a NASA archive where the plant was to see if there was a toxicity or anything like that, which they found not to be the case. Also, we have plants that have sprouted on the moon. The Chang'e 4 mission from China sprouted cotton seeds. The seeds grew upwards, which suggests that 1/6 gravity is at least enough for the plant to orient themselves and begin to grow. This did not have survived the night capability, but it's a promising result. Oh, kind of with that background out of the way, I want to move into my research, right? So when I think about this, there's lots of categories that we need to think about. We need to think about soil structure and aggregates. The moon basically has none. Water interaction. Anyone who's played with Lunar Regolith simulant and add water to it finds that it does not behave like dirt. It does not behave really like anything you would expect. It's extremely hydrophilic. It becomes very, very tightly bound to each other when water's first added, having this kind of hard pan effect. There's lax cation exchange. The soil can't really hold nutrients because there's no clays. Whatever nutrients that are in the regolith are not available. Organic matter is basically absent. As far as we know, microbial life and fungi are absent on the moon, though I'm not convinced they're totally absent from the moon. pH is actually not really a major concern, and particle distribution we can solve with geological screening using just filters and that kind of thing, so could be overcome. But yeah, so this build comes into my research pipeline. I'm working on several papers that I'm working to finish up and submit for publication. But primarily, I'm looking to how to physically weather things using worms and microbes to ultimately introduce pioneer crops that can lead to production crops. So this is building off last year's presentation. I have a lot more of the results in. But yeah, so this is informing work, right? So I'm using earthworms. That's the primary thing that I'm doing. And based off research on Earth, we find that physical alteration, worms have gizzards. They can grind the particles in their stomach. Worms are known to chemically weather certain kinds of minerals, particularly feldspar, which I mentioned earlier. Regolith has a large component of. Worms have a biological colonization effect as they carry microbiome in their gut. Their castings create a soil structure that behaves much more beneficially towards plants with water. We expected certain elements, particularly iron, to become more available. We expected water retention interaction to improve, and perhaps some slight changes in pH. So when we talk about worms, there's three main categories. They're not discrete, but they're pretty good. Epichick worms live in leaf litter. Endogic worms live primarily in the soil. They never come out. They eat minerals in the soil. And anisic worms kind of come up, grab leaves, and come back down into their tunnel. They kind of eat them like a sarlacc from Star Wars, I guess. But I'm using red wiggler worms primarily. Red wigglers are epigic worms with some endogic properties. They like to kind of go up and eat, but they also will go down and burrow some. Some of my early research was around building a habitat, understanding how the regolith interacts with worms. The raw regolith will kill worms. It will dry out their skin. It's so dry. So the regolith needs to be properly hydrated and mixed together into the various components to understand how it works. Early research was with night crawlers. Those are the anisic worms. I did get them to grow for at least a while before I harvested the dish. They're very strong, large worms. They could break up its media. But it is important to note that night crawlers struggle to be commercially grown on Earth. Almost all night crawlers in the world that people use for fishing come from just south of Toronto. There's farm fields there with lots of cows, and people go out and pick up all the worms because for whatever reason they're not super interested in being grown commercially. So that led me to Escherichia fetida, or what we typically call red wigglers here in the U.S. And so these were kind of my initial prototype experiments. I combined the regolith with horse manure. Horse manure was used as a substitute for human manure. Horses are monogastric. They're relatively similar in digestion, and they don't require a tremendous amount of paperwork to conduct university experiments with. So that's what I went with, composted manure. I mixed it at a ratio of 70% regolith, dry regolith, with 70% or 30% dry composted manure. I mixed it all together and added water. You can see here it binds together, and my next slide will show But yeah, and then I put paper towels on the top. They wetted the paper towels. That simulates leaf litter. It allows the worms to be able to get out of the material if they want to, which they enjoy. But yeah, within 30 days, right, we go from basically moon dust on the left to, if I didn't tell you that was simulated moon dust, you probably wouldn't know. The worms go in, they start driving and breaking things apart. They're looking for the manure and the microbes to eat. Their castings begin changing the overall structure of the soil, and so on and so forth. Also did it with the mare. The mare, you can see here, this is a good picture of dry, the mixture. It has some clumps. Those are, I think, from the composting kind of getting powdered. But you can see when you add water, it immediately just solidifies and the worms cannot go in at all. Over time, the water will, and those bonds, I think it's the van der Waal forces, personally, will relax a little bit and the worms will eventually make their way in. So that's so on and so forth. By 30 days, they pretty much mixed everything over. By 45 days, it begins to smell like soil, right? So that smell where you walk out after rain or like good compost, it's starting in there. That's indicative of the microbes taking place and being happy. And yeah, by 72 days, that's somewhat a fixture of the light, but the color changes. Everything's been worked over many times, and as I have systems that have been running for over a year now with no problems, so they're sustainable. I also have noticed reproduction. It's kind of hard to find baby worms, but I found them in both the mare and the highlands type. So this is potentially self-sustainable, at least on Earth, depending on worms and lower gravity. Here's some electron imaging, just so you can see up close. The mare and the highlands. What I want to call attention to is the very sharp geometry of the various particles. This is due to their unweathered nature. And as we move through castings harvested from different days in my experiment, we can see that these particles are all grabbed together. And then when we begin to zoom in, we start to see somewhat subjective changes to the geometry, right, where we see less sharpness in these angles and more rounding. And as the days go by, we see the incorporation of the organic matter, which we would expect, as well as the softening of these particles. By 7 days, large sticks and twigs and things like that are being digested, relatively large, are being pulled into the worm, and then being sent out with the castings, right? And we can confirm that, this thing that looks like a stick is in fact a stick using spectroscopy. And after 30 days, right, we can zoom in and we can see, we have a lot of rounding of these edges. We're no longer seeing those sharp particles that we would expect to see in the beginning, which is all positive for plant growth. For pH, maybe I skipped this bit. I ran a controlled trial for 30 days using 200 grams of media brought to 30% moisture, and then allowed the worms to do their things. And I harvested at certain dates to see them over time, the effect. pH doesn't change much, but pH would not expect it to. It does trend a little bit towards more towards neutral or 7 in the highlands, but highlands is around neutral to begin with. With the mare, we do see a trend somewhat down over time compared to the control, but that, again, not a huge result. Soil testing is where we get really strong results. We see in the highlands that nitrogen levels go down, but ultimately improve. And then we see carbon levels improve over time, which was not something we expected. And perhaps most strikingly is the cation exchange capacity, or the ability for the soil to hold nutrients that make them bioavailable, improved by 25%, which was a dramatic result in a very short amount of time. Mare saw very similar behavior, though instead of having carbon go up, we just saw that carbon decreased less over time with the presence of worms, suggesting the worms are helping sequester or lock up that carbon, which is interesting. We also saw that improvement in cation exchange capacity. Did a lot of testing around extractable elements. In the interest of time, I'll kind of blitz through it. But we expected iron to go up, but we didn't get a super strong result there. But in Mare, we saw improvement of boron availability. Highlands is where we saw a lot more availability. We saw aluminum become available, calcium, copper, potassium, manganese, nickel, and zinc. All suddenly were becoming bioavailable and detectable using assays. Here's just some comparisons, right? So in iron, and this is the highlands, we can see the worm dishes tended to have more iron available, but again, is it statistically significant? Probably more tests would need to be done, though it is a maybe 10-20% more of iron availability. Manganese actually was more pronounced, which was interesting, except at the end, after 30 days, less so. But that perhaps suggests that the nutrients that were available were in fact being brought into microbial life or things like that. Geologically, manganese and iron are substitutable. They behave very similarly, so interesting result there, worthy of further investigation. Perhaps more obvious of a result is around chemical weathering, right? So previous research done by Carpenter et al. Found that they studied worms just with regular earth soil to see if they could weather the rocks. And they found that relevant for us, they studied olivine and anorthite, which is the feldspars. Olivine they found did not weather, though they're not sure if it's inconclusive whether over a longer period of time of 6 months worms could ultimately weather them. But the anorthite or the feldspar, they found signs of weathering within 1 month. Right, so what we have here on the right here is a spectral signature comparing anorthite with worms and anorthite without worms, and a shift of those peaks suggests that the chemical substructure of the mineralogy is changing. So I repeated that experiment, and here I compare a day 1 highlands control versus a day 30 worm dish, and we can see many peaks shifting compared to the underlying the control. This suggests that the anorthosite, in line with Carpenter et al., is weathering rapidly, which is what we'd expect and indicative of chemical weathering and an actual change of the media. Mare, we saw much less structural shifts when we compare the dishes, but the Mare dishes are primarily pyroxene and olivine with some feldspar, maybe 25-30% feldspar. So whatever shifting is happening is being likely masked by the fact that most of the other underlying minerals are far more resistant to the weathering. But this is a very promising result, and when we think about food production, the fact that we can weather highlands minerals and become more soil-like quicker is actually helpful for South Pole habitation, particularly since the Mare material might be more valuable as a mining for ilmenite and titanium, as an example. Also conducted a bioinformatic analysis. There's a lot of technical jargon here that I'll skip over, but basically there was no bioinformatic data I found about regolith simulants or actual moon samples that I can find, and what I have found is that basically, yes, with the introduction of the compost and the worms, we see an explosion of microbial life. I'm still working through the computational aspects to identify which broad categories of microbes are present and how they change over time. But microscopic life is certainly more than capable in Earth-like conditions of thriving. So this is kind of, hopefully I'll present next year, right, is breaking down what, this is not my paper, but another paper, just doing a characterization experiments and seeing how things shift over time and what kind of things. But it's definitely there, getting very strong results. So much that the lab that did the sample said, like, your control had nothing, and then everything else had this huge amount of biological data. And I was like, oh, we'd expect that, right? Because it started basically absent of life. But when we tweak the settings, bacterial life just goes wild. Down the last few minutes, I'll talk about some plant growth stuff. I'm primarily looking, when I think about plants, a lot of people, like in the Plant the Moon Challenge or things like that, they just put a tomato seed in the regolith and then they get disappointed that it doesn't do well. I wouldn't expect it to do well, right? It's an unprocessed, unfiltered, unimproved rock. And so when I think about these things, I think about pioneering plants that can survive in those kinds of soils that are industrially useful, carbon and nitrogen fixing, or capable of producing biomass. If it has all these things and can be edible or medicinal, even better. That's led me primarily to look at dandelion. Dandelion's extremely useful. You can eat it. It grows everywhere. It's perennial. It can reproduce. It can reproduce on its own. It can be seen anywhere from Hawaiian lava flows to basically every part of the earth. You can make rubber from it, and it has known medicinal qualities as well. So yeah, I did a plant growth trial experiment spring of last year. and the worm dish, I'll cruise along. The worm dish did better than every other dish combined, right? And it was processing. The water was infiltrating. You see very healthy leaf color, suggesting that the nutrients are bioavailable. Whereas in the other dishes, they're showing signs of phosphorus or nitrogen deficiency, or their root structure becomes extremely stunted here in dish 2 plant 1, where it just struggles to move through the media, versus the worm dish here, dish 5 plant 2. It goes everywhere. It begins forming these things. We see these dandelions everywhere. These are photos I've taken on Hawaii and the Canadian Shield and Tobermory. Right, they grow, they're resilient, they're structural, and so I think they'll be one of the first things we should plant. And we can look at the root structure, right? We can see here with the worms, the root of the tap root of the dandelion is interfacing with the minerals around it in a healthy way. It's got a nice cone as it works its way deeper into the soil, whereas without the worms, it becomes stunted. The rocks just like seem to block it. Or without the regolith and simply just compost, dandelion doesn't do very well either, which is interesting finding. I have had some potatoes self-seed. The conditions were not right for keep them going, but promising as a potato crop. And I've also begun looking at other crops that might grow within the new lunar soil or the earth lunar soil I produced. There's some intellectual work property around this that I've submitted just as a disclaimer. And yeah, as I go forward, I'm going to work to kind of write up my results and quantify, really to help develop these specific technologies, really to consider scalability and flight readiness, and ultimately do more trials, right? This method could also be applied to Martian or asteroidal research, as that area is poorly understood at best. I'd like to thank my research committee and the people who've helped on this work. And yeah, I think that's about it. Thank you.
Nice presentation.
Thank you.
Good visuals and lots of good words also. Good backup.
Appreciate it.
Thank you for the comment. Any question? We have time for one.
I can just see the list of things that you're going to require to go to the moon, and somebody's going to ask you to justify why you need horse manure.
Yeah, so what we'd actually do, right, is I have a design of essentially a human composting toilet. We'd probably want to, my design, you'd want to process it, right, and then we could throw it in there. But interesting, I didn't mention, but worms are used in composting toilets around the world. They're known to eat human pathogens and viruses. So they are a way to sterilize the soil as well. Or that
well, waste. I would imagine that you probably would send up some worms that had been acclimated to using human waste.
Yeah. So which worms, and that's all like further research for sure. But it can be done. I believe.
Beautiful. Thank you.
Do you have any ideas how the lower, if the lower gravity on the moon will in some way influence the results?
Yeah, so that is the question. I don't know. We could, if needed, if say the worms struggled in 1/6 gravity, we could build centrifugal containers where it's a lot easier to create more gravity than to reduce gravity, right? So we could put this composter in a centrifuge or something like that if we really had to. But I don't know. No experiments have been done. No earthworms have been taken to the space station. They have taken flatworms, which aren't really the same thing. They did fine. I suspect worms can survive in microgravity or lower gravity, but that's an area of further research.
Stuart, very quick.
The simulant that you use, is there a place to get mass quantities of it that's close enough to the simulant? Like what region on the planet, what kind of dirt or... Because yeah. I'm thinking about, when to make a large scale study, you don't, like buying it from Exolith is prohibitively expensive.
Yeah, I don't know. There different places have attempted to set up simulant labs. There's another group, I think they're called Planetary Resources. They also produce simulant. I could double check and put it in the chat. But yeah, if you want to do a bulk order, your best bet might be to contact them and attempt to negotiate with a salesperson or something like that. I think the biggest problem is the shipping. So if you were to go with a truck or something, maybe you could make it work. But a lot of the companies a lot of universities have like built their own thing. Like, oh, they go out to the basalt fields, or there's a mine or something, and they, shave away or dig into the rock and then get the kind of the pure rock and powderize it. It's difficult, and a lot of those places kind of start. They go for a couple years, and then the funding dries up or whatever, and they go away. I went with Exolith because it's commercially available and other people could reproduce the work.
Yep. Thank you for that, Anna. And we